Hello, myself Gayatri Shravane, and today I am going to talk about gene regulation by miRNA. So, what is miRNA? These miRNA are small RNA sequences which are 22 nucleotides long, and these are a kind of non coding RNA which are endogenous in origin. So, what are the functions of miRNA? miRNA mainly functions in target protein silencing, which is a type of gene regulation of post transcriptional, and it regulates the target protein by target mRNA degradation or inhibition of its translation. How are miRNA biosynthesized? So, as I said, they are endogenous in origin. They are biosynthesized in the nucleus from the genes that are present itself in the chromosomes, and after that, post transcriptional modifications are carried out in the cytoplasm. These miRNAs are produced by RNA polymerase 2, and these genes are mostly present in the introns and exons of different genes. So let's talk about the biogenesis or the production of the miRNA in the nucleus. So the miRNA biogenesis begins with the transcribing a gene into a primary transcript. This transcription is primarily mediated by RNA polymerase 3, and some are mediated by RNA polymerase 2. Now, the step 2 in the process is these pre miRNA are cleaved by a microprocessor complex composed of RNA binding protein DGCR8 and type 3 RNase DROSHA into a 85 nucleotide stem loop structure called precursor miRNA, or shortly called as pre miRNA. So, here what happens is the stem of the hairpin structure is cut off and only a loop structure is remaining, which is called as pre-miRNA. So until the step 2, all this happens in the nucleus. But for other modifications, the pre-miRNA is transported from the nucleus to cytosol. Once the pre-miRNA is transported to the cytosol, an enzyme called dicer then cuts off the loop of the pre-miRNA and converts it into a double-stranded RNA, also called as miRNA duplex. The dicer itself unwounds the miRNA, and a single stranded miRNA is produced, which is also called as mature miRNA. miRNA is incorporated into a protein complex and is termed as RNA induced silencing complex. Now let's talk about how does a miRNA regulates gene expression. miRNA suppresses gene expression by usually binding at a site in the untranslated region. The gene regulation takes place by mostly two methods. First, target mRNA degradation, and second, by inhibition of the mRNA translation process. Now, whether the target mRNA would be degraded or its translation would be inhibited is determined by the degree of complement between the miRNA and mRNA sequence. If the sequence is fully complement to each other, then the RISC complex would cleave and degrade the RNA. But if the sequence between the miRNA and mRNA is not fully complementary, then it prevents endonuclease activity and only ends up blocking the RNA sequence for translation process, and hence there is inhibition of translation process and no synthesis of further protein. Some disease caused by dysregulation of miRNA could be inherited disease, which are caused by mutations and deletions of the miRNA. Dysregulation of miRNA also causes cancer such as colorectal cancer, lung cancer, and heart disease. Cancer can be caused due to two types of dysregulation in miRNA. First one is downregulation or loss of the miRNA, which functions as a tumor suppressor gene. Or the second one, which is amplification or upregulation of miRNA, which functions as an oncogene. In first type of dysregulation, some of the miRNAs that degrade or inhibit translation of tumor causing mRNA are lost from the chromosome or downregulated, which leads to proliferation of cancer. Some of the examples where loss of miRNA causes cancer: chronic lymphocytic leukemia, where miRNA 15A and miRNA 16-1 are deleted from the chromosome, as well as in lungs or breast cancer, miRNA called 7 miRNA proliferation of lung and breast cancer. Second type of dysregulation is upregulation of another kind of mRNA which suppresses the tumor suppressing gene, also called as the oncogenic miRNA, which leads to proliferation of the cancer. Now, some of the examples: oncogenic MIR21, which is found to be upregulated in numerous cancers. That was all. Thank you.